I've got sizes from small all the way up to extra large. So what we're doing today, just to kind of give you a little bit of a background, this is a simple genetic engineering transformation, and what that basically means is that we are giving a gene to a bacteria that this gene doesn't normally belong to this bacteria. It belongs to a different type of bacteria. And so just to kind of read you the little excerpt from this, just to give you a background on what that gene does. It says bacteria that produce light are very common in the ocean. Some types of bacteria that produce light are free living as plankton. Others live symbiotically with higher organisms such as fish and squid. The fish and squid hosts use the luminescence of the bacteria for several different purposes, including attracting their prey, communicating with others of their species, and confusing predators. However, it is unknown what benefit the glowing bacteria get out of this relationship. Perhaps you and your class can develop a hypothesis to explain why these bacteria have evolved to glow. One type of luminescent bacteria is called Vibrio fischeri. In nature, these bacteria live in the light organ of the fish Monocentrus japonicus. This Vibrio fischeri produces light through the action of the enzyme luciferase on a particular aldehyde. Because the reaction requires a great deal of the cell's energy, 10% or more, the process is highly regulated. There are several genes involved in Vibrio fischeri's light production. The luciferase enzyme itself is composed of 2 different subunits encoded by 2 different genes. Synthesis of the aldehyde also requires the action of several genes, as does the regulation. Together, all these genes required for the light production are called the Lux genes. The group of Lux genes from Vibrio fischeri has been placed on the plasmid, and that is the plasmid that we will be giving the E. coli bacteria that we're working with today. So basically, there are bacteria deep in the bottom of the sea that have the ability to be bioluminescent, and we are going to take that bioluminescent ability and give it to E. coli instead. So we're going to be making recombinant DNA. We're going to be making a genetically modified organism in that case. We're going to be using this plasmid with those genes as a vector for those genes. And in transformation, the bacteria simply take up the gene from their environment. And so I have the gene located on a plasmid, and the amounts that we work with are incredibly small. But I have the gene on its plasmid in this little vial, and I got to keep that on ice. Cold. And we are going to place it into a broth solution in which there's bacteria already in there. And just seeing that DNA plasmid in their environment, they will just take it up. So that's kind of a cool ability that bacteria have that not really any other organisms that we work with in the lab can do. So the idea is that when they take up that gene, since DNA is a universal language, they can read that DNA, even though they've never seen that gene before ever. Since it's their language, they know how to read it, and they can translate it and make the proteins that glow. And so we're taking a gene from one bacteria and giving it to E. coli. Along with that, and if you are interested in seeing the plasmid map, I've got a picture of it here. It shows what other restriction sites are located on there. If you've taken Gen Bio, you might remember what I mean by restriction sites. So you can see where we can cut it out with scrapbooking scissors, and we can glue it into other places. And there are some other genes that are located on there. One of the other genes that's located on the plasmid is ampicillin resistance. And so in this selection process, we allow certain bacteria to grow, but not all bacteria. That's why we have 2 different plates going on, because only the bacteria that have taken up this plasmid with this gene that we're giving them, those are only going to be the ones that are allowed to grow on the plates that we will set up. So that's why we got 2 different plates, because we will be selecting for the ones that actually worked. So that's a selection process. If you've taken micro, then you should be familiar with that. So that's the background of what we're doing. Mr. Khan is back there with a camera. He's going to be videotaping us. Anything you say or do can and will be used against you in the court of the internet. So be careful with that.
Not the first time I've been on YouTube.
So I will let you guys get your lab coats on, get some gloves on, and we'll go through with instructions. We'll get the lab set up, and we'll go through it. This should take the full hour. There's a little bit of an incubation period in the middle. We have to wait for like 20 minutes in an ice bath, and then we'll kind of walk through what's going on at each step too. But this is genetic engineering at its simplest. So pretty simple procedure. I think you'll be kind of amazed once we get done with it and be like, I just did genetic engineering. So it is pretty simple. So all right, we'll pause. I'll let you guys get some PPE on. What size is the lab coat today? So 15 minutes is up. So now that 15 minutes is up, it is time for the heat shock step. What we're going to do is we're going to take our beakers with our ice bath and our tubes in the ice bath where they have been incubating. We're going to take them over to the hot water bath in the corner of the room. It's at 42 degrees Celsius, and this is called the heat shock step. It will prompt the bacteria to take up the plasmid that is now in their environment. This is going to be performed for exactly 90 seconds, so one person needs to keep their eye on the clock. When they say go, make sure that you stick your tube into the hot water bath, agitate it around for a little bit. Ever so often you want to keep it mixed in that process, and then as soon as 90 seconds is up, it goes directly back into the water bath for recovery. That is going to be a 2-minute recovery that we're going to use this time. So I'll let you guys get set up to do that.
I'll see when. I want to do the agitation. I see if you're good at that.
Agitating?
Come with this.
Well, I gotta watch the clock.
Okay. Just let us know when.
Make sure it's at 42 degrees Celsius before you stick it in there. Is it still at 42? Yes, ma'am. Alrighty.
We're waiting on you.
I have a stopwatch.
Yeah. Okay, go.
Now, do we just bring that out at room temperature?
Should I get up there?
No, you stick it in.
Okay.
Get me nice and warm. Of course, if all people, that'd be me.
All right, we've got 2 minutes for this little thing to recover. So let's talk about the next couple steps. After this is done recovering in its little ice bath from the awful heat shock that you guys needed, we're going to give it some nutrients and encourage growth again. So the nutrients is going to be found in the Luria broth vials in your little ice water baths. And you're going to add in 250 microliters of that, so it's the same measurement of the calcium chloride that you put in from before. So the exact same way, you're going to measure it the same way, put it in there, mix it up. And the way that you mix it, you just use your finger to tap against the side, and that will agitate it enough to get it mixed up. So that's the next step that we do. Then we will let that come to recovery at room temperature, and instructions say 5 to 10 minutes, so we'll go a little bit over 5 minutes, maybe like 7 or something like that. So this will be the last kind of downtime, incubation time while we're waiting. After that recovery at room temperature, then we're going to be ready to plate it. So then we will be able to put a small amount of cells, 100 microliters worth of cells onto each of the 4 plates that you have. And then we will put them in the incubator after we get them all nice and spread out over the whole surface, and we'll give them about 48 hours at 30 degrees Celsius to incubate. So tomorrow we may or may not be able to see some glowing on the plate number, we said plate number 4 should have all glowing. We might be able to see some on number 3, so tomorrow at the very earliest. Otherwise, we should be able to see some pretty good glowing and good growth at 48 hours, so on Friday. So come back and see me on Friday because it should look phenomenal then. Let's see, so it's been a couple of minutes. Let's go ahead and add in Luria broth. You should have a sterile pipette in front of you. Add in 250 microliters of Luria broth. That's the same measurement of the calcium chloride.
Don't make me measure.
Nope.
There she pops.
I got her. Can't read the label now. You ever read the label on?
Can't really see the thing. Which one is 2?
Here. I got it. And then we leave it at room temperature, right?
Yep.
Should I make engineering?
Cute.
Okay, so we will give this 6 or 7 minutes for recovery at room temperature. In the meantime, I'll go over some plating with you guys before those 6 minutes are up. In the meantime, I'm going to show you how to do the little glass bead thing here. So these glass beads are sterile. And we're going to use these as a way to mix the bacteria, I guess not mix, but to spread them out across the surface so that they all have, you know, a nice even way to grow across the surface. So what we're going to do is you're going to have your little plate and you're going to open it up just enough to where someone can put in some beads. Be very careful when you do this because you could dump the whole thing in there, but just try to dump, 4 or 5. It says, I guess, 4 to 6 beads onto the plate. Also, be careful because they do tend to bounce on here too. So be very careful when you put 4 to 6 beads onto your plate. Once you got your beads on your plate, you're going to shake it back and forth like this to allow them to go back and forth like that. Shake it from side to side, allow the same thing to happen. What you don't want to do, you don't want to do this because it's not going to help to have your little beads around the outside going in a circle. You want them spreading this broth across the top of the agar so that it has equal growth. So we are done with our room temperature incubation. So at this point in time, we're going to measure out 100 microliters of the cells from the room temperature incubated tubes with your bacteria, with the plasmid and without the plasmid. 100 microliters, if you go back to the little diagram on the front page, it shows you where to measure that. It's the mark underneath the mark that you were using last time. It's half the amount that you were using last time, less than that. So you're going to measure out 100 microliters and put it onto one plate, just squirt it in the middle somewhere, and then also measure out 100 microliters and squirt it onto your other plate. As soon as you have it onto your plates, get the beads on there and get them spread side to side, back to back.
I'll do the beads like this.
When you are done with the beads, step 14, letter A, shows or tells you how to get rid of them, but you're going to clamshell, which means that you're going to open up your plate like this and allow them to just kind of trickle out, and I'll give you a beaker to put them in, and from there I will sterilize them in the autoclave.
I can't see it.
So your little broth is going to go onto each plate.
Where is it?
As soon as you have your 100 microliters onto your plate, and you're going to want a new pipette because you've touched the tip.
Oh.
It has to remain sterile. As soon as you've got your broth onto your plate, get some beads on there and mix it around. The broth will end up absorbing into the agar, so you don't want all of your cells to be in one spot. You want to get it mixed around as soon as possible.
Are we blind?
Am I actually looking for 100 or am I just kind of guesstimating it below that line?
It doesn't have it marked.
I don't think it does. I don't see it.
It's the lowest mark before it goes through the...
So,
Okay, so
Below
see where your
that?
250 is?
Okay,
It's,
that's 100.
That right there.
All spread.
okay, so it's the one below
Oh,
it.
it's
Uh-huh.
just that
Yes.
right there.
yes.
Okay, that's 250. The one below it is
No, get over
Oh,
here
I
and
thought
get
that was
beads.
250.
no.
That's 250.
That's 250.
So
That's where we need to be.
okay.
Okay.
I'm not
So-
looking for numbers printed on there.
They've got numbers printed, I think, at the 0.5.
Yeah, at 0.5.
Yeah, 0.5, but there's nowhere in that.
They've got the score, but they don't have that. So the
Yeah, I was
100.
looking for numbers.
So the 100 would be here.
Okay, your turn.
Yeah. Okay. And there's still a little bit in there.
Mm-hmm. Yep.
Sure does smell interesting.
Yeah.
Is
Okay.
that?
The gel.
Yeah.
It's supposed to clean gel. few more than what we need. That's
Keep getting too
okay.
much in there.
Yeah.
Do the thingy.
Do the thing.
Stupid bubble.
I know.
I can put the bubble out.
I can't get the bubble out.
The bubble's kicking my butt.
I could make a joke there, but we're being recorded.
Yeah.
For some reason, this sounds really familiar. Almost like the mini
Clamshell and
pinball
let it
game.
drop in there. Oh, you don't have your beads yet. Do you have neither one of them? Okay. Where's the beads in?
There you go. This
We
one.
have these.
Okay. All right, when you're done with your beads, clamshell and let them drop into here. Try not to get them to drop on the table, that would be bad.
Right, you heard them.
You might have to tap the plate. There you go. Come on up. All right. Tape up the edges.
Get out of there. You're sticking on the edge. How do you want the edges taped? Just all the way
Just
around?
tape all the way around so that you can't open the lid anymore.
Don't know how to do it on the web.
It's plenty.
I know what you're thinking of. It's those little puzzles with the beads.
Yes!
That's what you're thinking of.
That's what I was thinking of, because I actually have those puzzles in my brother's closet.
Can I have some tape, please?
There you go.
Thank you.
Like a nice little bacterial Christmas present.
Except these will not be opened again. So we're taping these up to get them sealed, so we're not going to be able to open them again once we've got a lot of bacterial growth in there. We don't need to get back into it again. So these will be incubated upside down. Being incubated upside down makes sure that there was not any condensation on the lid that would drip down and distort the colonies. So they get incubated in the 30-degree incubator, which is in the left corner. And the period of time that should be optimal at that temperature is 48 hours. So if you happen to be around tomorrow, come find me, come look in here. We'll see if there's any glowing. Otherwise, Friday, there should be the best viewing on Friday. If you come up here and look at it on Friday, you'll want to make sure that the room is completely dark. Go hide in like the darkest corner. The darker the room, the more bright that they will appear, the more easily that they will be seen.
I'll be here tomorrow.
Yeah,
I'll also be here tomorrow for your class.
yeah. We'll come up and look.
Your class is the only class I have.
No, I'll be here tomorrow.
Good. All right, 30-degree incubator upside down, 48 hours.